Hi guys, welcome to lecture 4 of our evolution unit. We're going to start by talking about phylogenetic trees and cladograms. So these diagrams, they're going to show the evolution of species, and sometimes they indicate traits of those species. They do have different looks, and you don't need to know the difference between the two, but cladograms typically will require you to add traits, whereas phylogenetic trees are more based on time. What is important for you to know, though, is that you must be able to use the evidence to draw cladograms or phylogenetic trees and draw conclusions about the relatedness of the species. So, as far as images go, this here is your tree. This is a cladogram. So let's talk about reading these trees and cladograms. So a branch, this is what is going to indicate that there is a common ancestor between different species. If we have recent branching between two species, it means that they are more related. Also, homologous structures are indicated. So all species have a downstream, all species downstream would have that structure. The species that is odd, the odd one out, that doesn't share the homologous structure with the others is called the outgroup. So if we take a look here at this cladogram, you can see it's a little bit different than the one up above. So there's many shapes that these can take. So looking at this, let's answer these questions. Are lizards more closely related to mammals or birds? So we find here, this is our lizard, and this is our bird. And then we have mammals up here. So if you look at this, they are more closely related to birds. Which species is considered the outgroup? And the outgroup, as I said, is the odd one out. It's not going to share any structures. And how we are going to figure this out is you can see these here are the structures that they've put in. So when we talk about the lizards versus the mammals, the lizards being related to mammals or birds, we can see that they do not share this amnion, but they do share right here, this feathers. So the outgroup would be the lungfish because they do not have any of the homologous traits listed below. Identify evidence that suggests that crocodiles are more closely related to birds than amphibians. So if we look at the crocodiles, our amphibians are way up here. And basically, we can tell that right here, that homologous characteristics. So there are shared traits. So let's look at an example. Use our knowledge of homologous structures to construct a cladogram showing the relatedness of the following species. Rearranging the table can make construction easier. So that is your first clue, is sometimes you will get these tables and they will have stuff all over the place. So these have actually been reordered over here to share the traits, to show the traits. Because as you can see, this yellow one doesn't share anything. So the right one has been reordered to make things easier. So there are two types that I'm going to show you. Both of them are correct. So we are going to start with the starfish. we know that they have eggs, but it's the only thing that they have in common. So we're going to draw this line here, and we're going to put this eggs right here. I'm just going to put an E for eggs. And this is going to be our starfish. So the next trait that we have in common is the frog and the eggs. We've got a backbone. So we know that the rest have the backbone, so we're going to start this. This right here is going to be our backbone. So after that, that's the only thing that the frog has in common. So we are going to branch that off here, and this is going to be our frog. Now we're going to create another branch for the next thing, which is going to be warm-blooded. And at that point in time, that takes us to the next thing, which would be the monkey. And the rodent and the bird, they all have that in common. And what they have in common is hair. So the only one that doesn't have that is the bird. So we're going to take the bird over here. And off on this side, we're going to add that other trait, which is hair. And we are going to go off each side. And we've got the monkey and the rodent. So that is one way to draw the cladogram. Another way to do that is like the first example that we saw. Draw that straight line. And our first trait is going to be the eggs. So here would be our starfish, since eggs is the only thing that it has. Then we would add our backbone. And next, as we did before, we will have our frog. And then we add the next trait, which is warm-blooded. And then that takes us to our birds. Our next trait is hair. And we would put here the rodent. And at the end here, we would put our monkey. Honestly, it is a matter of personal opinion which one that you use. I personally think that this cladogram is much easier to do than this one, but again, that's your choice. So example 2, the table shows selected characters used to determine relationships among 4 species of mammals. Construct a cladogram to show the relationship between those species. So the first thing you need to ask yourself is, do you need to rearrange? And we do. We can see that there's some that don't have the same traits. So I'm going to start with tail. They all have the same thing, and you could do it the opposite way if you wanted to and start here with just like the hair tufts. But I'm going to start with the tail. And then the next one I would do is the 3 toes, because 3 of those have that. And then we've got 2 with the incisors and 1 with the hair. And then you're just going to put the animals or the species in, and I'm just going to abbreviate. So it would be P, S, C, and N. Because we know that all 4 of them have this. These 3 have the toes. these two have the incisors, and this one has just the hair. So now that we've done that, we can start our cladogram. So the first thing that we're going to put down here is our trait, and this is the tail. And then off of that, we're going to draw our line. And the first one would be our pseudophilus. So I'm going to just put P there. The next trait that we have is 3 toes. So our next one here would be the spermocyon. Our next trait is going to be our incisor. And our next species would be the Castoropus. And then finally, we have the hair, which means that here at the end would be the neo. Okay, so our next example says draw a cladogram for the information summarized in the table. So as you can see, we have a lot where they have the same things. So this one's a little bit more difficult. So the first thing I'm going to do is I know that I'm going to need a big cladogram here. So I'm going to draw my big line. And I'm going to look back up here. What do all of them have in common? They've got the cells with the flagella. So that's going to be our first spot. Cells with flagella. And then we would just go through. And the only one that has that, just that trait, would be the sponge. So here would be our sponge. The next trait that we're looking at would be symmetry. So we're going to write that here. And our next animal that we're looking at is the jellyfish. Then we have another trait, which is the bilateral symmetry. So we'll put that here. We'll put bilat sim. And that is going to be our flatworm. And then let's go back up and look here. What is our next trait? The mesoderm. So that would be our earthworm. So mesoderm. And we're going to put earthworm. Okay, so that is the last one that has just that trait. So we are going to look at this section here where the head develops first, the anus develops first, and the segmented body. So what we're going to do is we're going to skip a couple of those for a minute. The next one that we're going to put down, the next trait, is the anus developing first, because that goes beyond this here. So we're just going to put that anus first. And with that, we've got the starfish and the human. So the first one we're going to do is our starfish. And then the last one that we would do would be human. And also, let's go back up here. We know we have a human. We have a vertebra. So we're going to put that trait in. And now I'm going to go back and do the ones that I have to fill in. So for instance, my starfish also has a water vascular system. So we are going to put that trait off of the starfish. Now, let's go back up here to what we skipped. So we skipped that the head develops first, and we skipped the segmented body and the calcified shell. So what we're going to do is we're going to go back to this earthworm, and right here we're going to put head first, because everything off of this, the head develops first. And then here we are going to put the segmented. So the head first covers the earthworm, the snail, the fruit fly. The segmented covers the earthworm and the fruit fly. So what we need to do is we know that we need to put in the fruit fly and the snail. There is another trait that we haven't talked about, which is the fruit fly. It's got a type of exoskeleton, so we know that we need to put that on there. So segmented, the snail is not. So we're going to put the snail right here between and we are going to put the fruit fly up here. So we have, with the snail, it's got a shell. So we're going to put that here. And the fruit fly, we know it has that exoskeleton. And that covers everything that we have. So these are not the easiest things to figure out. So make sure that if you are confused about those, try to do it on your own and then come back and look at it. So just keep practicing those. Now let's move on to micro versus macroevolution. So microevolution is the study of evolution at the population level. And we know that populations are the smallest unit that can evolve. An organism cannot evolve, but a population of organisms can evolve. So basically what that's saying is that a single individual cannot evolve, but the population as a whole over time will. Macroevolution is studying the evolution and changing of species over a long period of time. So with macroevolution, the population is a group of similar organisms that are all within the same habitat. And of course, habitat is their home. Species, the biological definition, is a group whose members, one, they can mate in nature, and two, they can produce viable or normal offspring, and three, those offspring are also fertile. So let's look at some examples. Donkey and horse, are they the same species or are they different? And the answer is no, because if you put a donkey and a horse together and they mate, then they create what is called a mule. And a mule is actually infertile. So they are not in that definition. A beef cow or a dairy cow. Let me go back here, highlight that one for you. A beef cow and dairy cow, they are going to be the same because it doesn't matter if they're beef or dairy, they could mate. Tiger and lion. Well, we tend to think of them as the same species, but they are actually different because they are on different continents. So, essentially, they can't mate in nature. And then a collie and a boxer. Well, both of them are dogs, and they can mate. As you know, there are some very interesting combinations of dogs in our world. Let's talk about speciation. So this is the evolutionary process by which reproductively isolated biological populations evolve to become their own distinct species. So basically what that means is we get two groups that are reproductively isolated from each other. They will, one, will not mate in nature, or two, will not produce viable offspring, or three, will not produce fertile offspring. So for instance, right here, we've got these fruit flies, and some sort of speciation event occurred right here so that this fruit fly became its own distinct species. Then we have another one here, so we end up with two more distinct, and they will not mate with one another. So what could have caused speciation? So some could have been born with a mutation, and because of that, other fruit flies may not recognize them, and the ones with the mutations mated, and then, of course, they had babies with mutations. Some could have been born with the wrong number of chromosomes, so if two flies, both with an aneuploidy, mated, it could lead to offspring with completely different number of chromosomes compared to the original, and only those that have that aneuploidy can produce offspring that are viable. Some could have been separated from others, and over time, in different environments, selection led to different phenotypes. And then the flies from the separate populations do not recognize each other to mate. So, speciation, there are two types. And those two types, we have allopatric, or allopetric, depending on who you speak to. And then we have sympatric. So the allopatric speciation is when a population basically becomes separated over time due to a geographical barrier. I can't spell. So for instance, a river, a mountain, a highway, the barriers basically restrict the gene flow, and then the population becomes more diverse, leading to population members that can't recognize signals from others. And keep in mind that this usually takes hundreds of thousands to millions of years. So when you think of this allopetric, think of a mountain. Think of that A as a mountain, and that will help you remember it. And this diagram basically shows you what's happening. You start with two populations that are the same, and then that barrier interrupts the gene flow, and you end up with new variants in each side. And then basically, eventually, you end up with those two different species. So, sympatric speciation is when a population develops into two or more different species while being in the same area. So when you think of sympatric, use that S to think of same. So the sympatric speciation due to mutation is one of the causes. So for instance, in plants, this sympatric speciation occurs frequently when a plant is produced that is a polyploid. So basically, it can self-fertilize and it can make many offspring that can mate. But polyploids cannot mate with diploids. So what happens is we end up with reproductive isolation, and that means that we now have a new species of plants. So it can also happen without mutation. So for instance, there are fruit flies. They fed on hawthorns, which are apple-like fruits. And then the apple tree was introduced by man, and some of the flies preferred the apples to the hawthorns. And because of this, it led to differences between the populations until reproductive isolation occurred. And then we ended up with 2 different species in the population. So there are reproductive isolating mechanisms. These are also referred to as RIMs. For speciation to occur, the reproductive isolation must occur. And then that reproductive isolation is going to lead to the maintenance of 2 different species. And here are some examples of the isolating mechanisms. So there are 2 main types. We have the pre-zygotic and the post-zygotic. So the pre-zygotic reproductive mechanism stops the formation of a zygote. And a zygote is that first cell that's formed after fertilization of the egg occurs. So there are different types of the pre-zygotic RIMs, which I spelled that wrong up here. I apologize. I was thinking of RIM, sleeping RIMs. Okay. So the different types of RIMs, there are geographical isolations. And this happens when 2 species are never near one another geographically, and they never meet and reproduce. So for instance, we've got here in North America, we have rabbits. In South America, they have these hares. Well, due to geography, they're never going to meet, they're never going to reproduce. Then we have what is called temporal isolation. And this is when 2 species breed during different seasons or times. So for instance, the eastern spotted skunk, it likes to breed in the winter, and the western likes to breed in the summer or the spring. Another kind is behavioral isolation, and this is when the behavior of one species is not recognized by members of the other. So for instance, sounds, calls, there could be pheromones, courtship rituals, any of those not recognized. Another kind is mechanical isolation. And this basically is just structural differences that prevent successful mating from occurring. And then the last one is gametic isolation. So this is when the sperm of one may not be able to fertilize the egg. It might not survive in the reproductive tract. And there could be lots of different reasons for that. pH, different things like that. So let's talk about post-zygotic reproductive isolating mechanisms. So these mechanisms are going to occur after the formation of the zygote. So the types of post-zygotic RIMs, the first one is zygote mortality. So basically the zygote is not viable and it ends up dying. Or there could be a zygote with 2 different chromosome sets, and it may fail to go through mitosis and develop any further. The next one is called reduced hybrid fertility. And this is when an organism develops but cannot reproduce itself. So for instance, we talked about the donkey and the horse equals a mule. That's what's happening. So they cannot produce normal gametes through meiosis. They are actually sterile. So let's look at an example question. 2 species of frog belonging to the same genus occasionally mate, but the offspring fail to fully develop and hatch. What is the mechanism for keeping 2 frog species separate? So A, the post-zygotic burial called hybrid breakdown. Maybe. The pre-zygotic barrier called hybrid sterility. Well, we know it's not that because they keep having offspring. Gamete isolation. We know it's not that. And post-zygotic barrier called hybrid inviability. That one makes the most sense in our case. Okay, so let's look at types and patterns of evolution. So we're going to see many patterns in the fossil records. Let's go through and we're going to label these graphs really quick so that you know which one goes with what. So this one is called divergent. This is a parallel pattern. This one is an adaptive pattern. And this is a convergent pattern. Okay, so divergent evolution is occurring when speciation has occurred, whether it be allopatrically or sympatrically, and the 2 species are going to continue to develop different features, and they will end up becoming more diverse or different from one another. They will possess homologous structures that indicate a common ancestor, though. The parallel evolution occurs when divergence occurred and the 2 different species evolve similar adaptations, usually because they're in the similar environments. Then we have adaptive radiation, and this is occurring when what is called rapid diversification occurred in a short amount of time from one ancestor. It often occurs when 1, new niches become available as individuals are relocated into new areas, or 2, when a drastic change in the environment causes a mass extinction and opens up new niches. So, for instance, this one is adaptive radiation. These are actually the finches on the Galapagos Island. And as you can see, they all started out as one type of finch, but then they were separated when the islands formed. They were in different areas, and because of that, there was different food sources available. So you can see that they all started out kind of similar, but there's some with small beaks, there's some with big beaks, there's some with very skinny beaks, and all of that is adaptive based on where they're at. Convergent evolution occurs when 2 species have similar features, but they are not related. So the similar features that evolved independently in each species, these are going to be called analogous structures. So an example of converging species would be sharks and dolphins. They're very similar. Birds and bats, pill bugs, and the pill millipede. So let's look at this really quick. Divergent. And we can draw a little arrow here for that. And convergent is going to go here with this box. So basically, divergent is going to lead to homologous structures, similar structures in a different species due to a common ancestor, versus convergent is going to lead to analogous structures in different species that are not closely related because of their environments. So analogous structures are similar structures to serve a similar function. And the following are some examples. So the first one is the flippers of penguins and the fins of fish. They're similar, they're both on the outside, because they live in a similar environment and they have a similar purpose, which is maneuvering in the water. Another example is the dorsal fin of dolphins and sharks. They are very similar, but they are still different. And obviously they're different species. Another example is the wings on insects and the wings on bats. They're very similar because they live in a similar environment and they have a similar purpose of flying. So if you look here, keep in mind that internally the structures are not similar. So they are analogous. They look similar, but internally they are not similar. And that is going to bring us to the end of Lecture 4. And next time we'll start talking about coevolution. See you guys in class.